Everybody, my name is Professor Reinhard, and welcome to Psych 2. I will be your professor for the next 6 weeks. It's going to be a lot of information. It's going to be fast-paced, but I think we can get through it if you just keep up with the tasks as we're going along through the class. So today we're going to cover the introduction section and part of chapter 1. And then periodically I'll have you pause the video, and then I would like you to do the activities in the PowerPoint slides. So you might want to pull up actually there'll be Google slides, sorry about it. So you pull up the Google slides and have them ready so you can work along in the slides as we're going through the PowerPoint. All right, so first off, I have to share the PowerPoint, and I have a checker here because I recorded the lecture without sharing the PowerPoint already. So can you see the PowerPoint?
Yes.
All right, great. So I'm going to start, and Linda, you can log off if you would like to. All right, so as I already said, I'm dividing up the lectures. Every week we're going to have 2 to 3 chapters, but I would like you to just focus on one chapter at a time. Try not to do all of the lectures and all the chapters at the same time. So day 1, doing a little bit of introduction and a little bit of chapter 1. And what I want you to do first is go to your book and read page 4 and 5. And this page is it's about some of the mysteries that remain in science and some of the mysteries in psychology and neuroscience. After you finish reading it, I would like there's going to be a link on this page. I would like you to go to the Flipgrid, open up the link, and I want you to record a video of yourself. And in that video, I want you to introduce yourself, tell us your name, maybe your major, maybe an interesting fact about yourself, and then tell us what your thoughts are after reading pages 4 to 5. So go ahead and pause the video, complete this task, and then you continue the videos after you finish that. All right, so some of the mysteries that remain in psychology and science and neuroscience is, one of the big ones is this idea of consciousness. So we know that there are some things in the world we're conscious of. We have conscious perception of them, and there's many things that we're not conscious of. So one of the questions is, what is the advantage of consciousness? Why do we have consciousness? Another question is brain plasticity. So we know a lot about how the brain changes in response to experience, but there are many things that we're still trying to learn about it. Creativity and emotions. These can be things that are difficult to study with animal models, so there's still a lot of kind of mystery around those. Attraction. There are a lot of studies that have looked at attraction, both the neuroscience side of it and the psychological side of it. And it's very difficult to determine what factors play a role in how we're attracted to each other. There are also how humans make decisions. This is a big area of research. And the impact of our social world and the culture around us on our brain structure and brain development. So these are areas that are still kind of mysteries about the brain. And I would like you to think about some things you're curious about. So what do you want to know about how our brain works, or what are like the neural substrates of certain behaviors? Like right now in our country, we're dealing with a lot of fear over the pandemic, anger over the way people have been treated historically in the country, prejudice and racism. So are you curious about why some of these things happen? Are you curious about how maybe anxiety spreads in a culture, or human behavior, wearing masks, not wearing masks? What's the difference between who does and who doesn't? And so basically all of human behavior or animal behavior is kind of linked to and driven by the biology of the brain. So as we talk about biological psychology, which is what this text and this class is focusing on, you can distinguish biological psychology from, say, neuroscience or medical science is focusing on brain function and activity based on the focus of the science itself. So if the main goal is to relate our biology or the biology of the brain to issues of psychology, then you're probably talking about biological psychology. Some people who study neuroscience, they can spend their whole career just studying one little protein. Some proteins do an amazing number of things, and how they interact with other things in the brain is you can literally spend an entire career just studying one protein. Or some researchers that are focusing on neuroscience may be just interested in a particular organism. Maybe they want to know about camouflage in goodness gracious, squids and octopi, or maybe they want to know about differences in the nervous system in octopus versus humans or something like that. So there are many different possible focuses when you're talking about the overall field of neuroscience, but biological psychology is going to be focusing on how does the biology and function of the brain relate to psychology. The actual book definition is the study of the physiological, evolutionary, and developmental mechanisms of behavior and experience. So this is a very dense definition. And basically they're saying that there's different ways that we can define biological psychology. So you can focus on the physical structures, the evolutionary advantage that structures may have, how structures develop, the mechanisms that underlie behavior, and mechanisms that underlie our subjective experiences. And so these are all encompassed in the definition of biological psychology. And again, the focus is on brain activity and how activity between cells in the brain can produce an enormous wealth of behavior and experience. So this kind of essential question of how does just like neurologic impulses between cells, how does that allow us to have this feeling of self and reality? So it's kind of one of those fundamental mysteries is why is just interaction between cells generating our awareness of the world around us? So there are different ways when we're talking about trying to understand mechanisms of behavior and experience. There are different ways we can describe and define a behavior. So if you have the question, why does a bat fly? Why doesn't it crawl or walk or something like that? You could explain this behavior based on the physiology, which is body parts and the machinery of the body. So arms, limbs, maybe cells, things like that. So when you're talking about why does the bat fly, you could maybe focus on the wing structure itself and the air currents that are generated by the bat when it flies, and how those air currents allow the bat to be able to fly. You could focus on sensory cells in the bat wing that allow the bat to sense whether it's upright or to the side, and allow the bat to sense if it's generating the right type of air current. And these are areas of study that people are currently studying the mechanisms of bat flight. So that would be, those are physiological descriptions of the behavior or explanations. There can be ontogenetic explanations. Ontogenetic means the genesis or the origin of something. So this is describing how a behavior develops, including the influence of genes, nutrition, experience, and interactions. So with our example of bat flight, you could maybe study the actual development of the bat wing and compare that to the development of a mouse arm or something like that. So you're studying the actual muscles and the parts of the body, how they develop differently. You could also study the gene expression. So here we have gene expression in forelimbs and the hind limbs. These are the different genes, gremlin, FGF8, and SPRI2. Excuse me. And so you could study the expression of different genes in different body parts and try to understand what genes lead to the development of a wing versus a hand versus the webbed foot or something like that. You can also explain behavior using evolutionary explanations, which is reconstructing the evolutionary history of a structure or behavior. And essentially what you're doing is you're comparing structures or genetic expression between different animals, and you're trying to track if you see similar structures in similar animals across time. Basically, the more similar the structure is, the more closely linked those two organisms might be on an evolutionary timescale. So they're trying to see how did structures mutate in different animals across time. And you can do this by looking at something like the phalanges in animals. So you can, this would be creating a phylogenetic tree. So you're looking at body parts and structures, and physiological structures in animals. And you can compare the structures and see, you can construct a tree to kind of estimate how closely linked two species are on an evolutionary timescale. Or you can use gene expression. You can see how much of genetic DNA is shared between species, and how genes and the expression of genes has mutated and changed from organism to organism over time. So these are different evolutionary explanations for why a bat potentially could fly. So for instance, this is showing the bat is expressing gremlin in between the phalanges and the forelimb, whereas the mouse is expressing BMP, and the duck is expressing a combination of BMP and gremlin in kind of an interesting pattern there. So maybe they're saying the pattern of genes could explain how you have a webbed foot versus an actual wing versus phalanges without any webbing in between them. Okay, and then finally you can also have a functional explanation. So this is why structured behavior evolved the way it did. So these are kind of hypothetical, and you look at the context in which an animal lives, like its ecosystem that it lives in, or its ecological niche that the animal takes up within an ecosystem. And you can use that to try to explain what was the advantage for this animal to have this trait or behavior. So bats have a lot of very interesting adaptations. Of course, they fly. So they're the only animal, the only mammal that can fly. They're also nocturnal. And they're auditory specialists, so they hunt and navigate using sound. So these are all specializations of bats, and you might ask basically what's the advantage? So the advantage of a small mammal to be able to fly might be they have different food within the ecosystem they have access to versus just walking on the ground. Maybe they can also avoid predators when they're flying, or very specific kinds of predators, more so than when they're walking on the ground. So these are possible functional explanations of the flying behavior of a bat. Okay, so now again, I want you to pause the video and go ahead and please open up the Google Slides, and you're going to look at this leafy sea dragon. If you want, you can look up extra information about it online. So it's a sea dragon that's related to seahorses, and it lives in kelp beds. And it tries to mimic the movement of the kelp by drifting very slowly in the ocean and tries to blend in with the kelp. So over here, you should be able to directly edit the slide when you open it. So you can click in, there's a text box located here, which you can't see, but you just click right here, and you should be able to type in and directly edit the slide. So please go ahead and type in your explanation. What's a physiologic explanation, functional, ontogenetic, and evolutionary explanation for the behavior of the sea dragon here? Okay, so as we are going through the book, you want to keep in mind some key principles throughout the book. One of them is that perception occurs in the brain. So essentially they found through people that have brain damage that neurons and the activity between neural networks is what's actually generating our kind of subjective experience of self and the world and reality. So if you don't have a set of cells, say that if you don't have the cells responsible for processing color, then you won't perceive color in the world. If you don't have the receptors that are activated for color, you won't be able to see color. You don't feel it or experience it if your brain is not capable of interpreting that information. So perception of the world around us, our experience of reality and self, is actually something that's being generated in the brain. The second is similar to the first principle, that the mind does not exist separate from matter. So I'm not, people have their own individual religious beliefs, and completely respect that, but from the scientific perspective, there's not a separate, there's not like a human mind or entity or spirit that's separate from the biology, which is our brain. So our sense of self and reality depends on the biology, and the biology produces our sense of self and reality. So without the parts of the prefrontal cortex, we may not have our same personality. So who we are is in part determined by our biology. And then the third point is to always be cautious about assuming causation. In science in general, and this is true in psychology, it's true in neuroscience and any kind of science, is you always want to be looking for alternate explanations, because a lot of times, even when you do a very careful, well planned out study, you're not necessarily proving causation. It's very difficult to definitively prove causation of two things. So say if you, this is kind of what we're more used to in psychology studies, is maybe they find depressed people also tend to be less active. But does that mean that being less physically active leads to depression or causes depression? Or does that mean that being depressed leads to less physical activity? So the causal factor between these two variables is not clear, and we can't make a solid claim about causation. A study that might seem more scientific maybe is that maybe genes, they found that certain types of genes tend to be more commonly expressed, either upregulated or downregulated, in people that have depression. So this seems more scientific because we're talking about genes, but still the same thing is true. So does this mean that depression causes, say if the genes are decreased in depression, does depression cause the decrease in these genes, or does a decrease in these genes cause depression? Again, it's not clear. And it's similar to the final example. If they find certain brain areas are less active in people with depression, again, does that mean that the brain area leads to depression, or depression leads to decreased brain activity? So always be cautious about claiming a causal relationship between two factors. One of the best ways to kind of get closer to proving or supporting a causal relationship between two things would be if you can show necessity. Wish there were a way to type on the screen instead of draw on it. Necessity and sufficiency. Sufficiency. Okay, spelling is horrible. It's gonna be horrible always. It just is horrible. So hopefully you get the idea. So necessity and sufficiency basically means if, okay, for instance, we talked about color. If I think one brain region is causing our ability to see color, I could maybe find that brain region and stimulate it. So I'm going to activate the brain region, and I'm going to see, does this person experience color? Do they have a perception of color? So if that happens, I prove sufficiency. So this brain region is sufficient to produce the perception of color. Now the opposite, if I take out the brain region, or if I somehow decrease the activity of that brain region, and I see the person can no longer see color, I've now proved necessity. So I'm saying this brain region is necessary to see color, and it's sufficient to produce a perception of color. So if you can design an experiment that shows necessity and sufficiency, it shows, it gets you a little bit closer to this idea of causation. However, keep in mind that within the brain, within neural network systems, usually there are many brain regions working together. So where one brain region might be necessary and sufficient, it doesn't mean that there aren't also other brain regions that are also necessary and sufficient. Okay, so I'll stop there and keep going here. All right, so we're going to get into chapter 1. And we're just going over some of the basics about neurons, brain biology, cells, and things like that. All right, so the first thing to kind of wrap your mind around is just that we have a lot of brain cells. There's a lot of cells packed into the brain. So throughout the rest of the lectures on neurons, oftentimes you're going to see a slide with like one cell on the slide. And it kind of gives you this perception over time of like a cell by itself hanging out in a bunch of space. But in reality, that's not what's happening. So in these images, all the tiny little circles are all individual cell bodies. You can see the same thing here. This is a line of a bunch of different cell bodies. Here is a layer of other types of cell bodies. And this is a close-up shot, so this is more zoomed in here. And again, we can see these small circles, the blue circles here, are different cell bodies. The way they create these images is just they can stain the cells with fluorescent proteins, or sometimes inject the cells with fluorescent proteins. And then you can image with certain wavelengths, and it'll generate these kinds of images. So the cells don't normally look like this. It's just fluorescent dyes we put in. But so the point is that there are many cells packed in together in the brain in a small amount of space. So in 1 cubic millimeter, which would be very tiny, some small space between my fingers, there are as many as 50,000 neurons. The exact count is going to be different for each person, different for male, female. And it's some of the studies trying to count cells are, it's a difficult task. So this is kind of a rough estimate. There can be more or less, but in general, a small millimeter of cortex has about 50,000 neurons. In the brain altogether, there are about 100 billion neurons. And then each individual cell, so say like this little green cell here, it extends its process, which is its axon. And these processes are how cells communicate with other cells around them. They send out kind of like arms and legs, and it's as if they're communicating through their fingers and toes to other cells, kind of, and sending out little signals that way. So one individual cell is contacting, has synapses with 6,000 other cells. So this one individual cell can possibly talk to or communicate with 6,000 other cells around it through these synapses, which are the sites of communication between cells. So basically, there's a lot going on in our brain, a lot of activity, a lot of communication between different cells. Neurons have different shapes. Another name for that is morphology. They have different morphologies, and the morphology of a cell is going to influence the function of that cell within a neural network. So on all of these, again, the small little circles are the cell bodies, and the branching parts of the cell are the processes, or the dendrites and neurons. So processes kind of means the extensions coming out of the cell, which can be either dendrites or neurons. Okay, so in this particular cell, you see from the cell body, there's only a couple processes. So these are axons, a couple axons extending, and only like 5 or 6, the synapses will probably be along here. So not very many regions where the cell can communicate with other cells. Compare that to this cell or to this cell, where it's going to be taking in information from the dendrites from a lot of different cells, and it's going to be communicating with a lot of different cells because it has all these different branches. On each one of these branches are regions where that cell can communicate with other cells. So a lot more possible communication in this cell, cell A or cell C, than cell D. Another neat thing to take note of, is kind of the length of the cell. So we'll talk later about the cortex has 6 layers. Cortex is the part on the outside of our brain, and it has 6 different layers. And we'll talk about that more in depth later on, but basically there are different types of cells that tend to be localized. They tend to live in different layers of the cortex, and all of those different kinds of cells will have different morphologies. So say, for instance, this cell, you can see that its cell body is maybe layer 2 or 3, but it can contact cells all the way from layer 1 to about layer 5. This cell is contacting cells again from all the way from layer 1 to about layer 6. However, this cell is only contacting cells in layers 1 and 2. This one is only in layer 2, 3. So you can see how the shape of the neuron is going to affect how many other neurons it can communicate with, and whether or not it communicates with local neurons in a small area of cortex, or whether it communicates with a lot of different neurons across areas of cortex. Okay. And then besides neurons, there are also other kinds of cells in the brain, and these are collectively called glia, or glial cells. Glial cells outnumber neurons by about 50 to 1, so there are a lot more of them. And it used to be the case that researchers thought that glial cells didn't really have a function other than to just kind of cement the neurons in place. So they just kind of held the neurons up where they're supposed to be. That was the idea. And so originally they're called glia because glia means glue. So they thought these cells were just kind of gluing the neurons in place. And now we know that each type of glial cell has a specific function, and the functions are very important for the proper survival and function of the nervous system. So there are 4 different types of glial cells. There are astrocytes, oligodendrocytes, microglia, and then there are Schwann cells. Each type of cell has its own specific function. Okay, so oligodendrocytes are these guys in blue here. And what they do is they take their processes, which is kind of like their arms or their legs, and they wrap their processes around the axons of the neuron, and they create what's called a myelin sheath. And we'll learn more about that later, but basically the myelin sheath helps the axons to communicate better and faster. So the oligodendrocytes create the myelin sheath around the axons. Astrocytes, we see them in green here. And you can kind of see what they do. So astrocytes are contacting the synapses of the neuron. And, excuse me. So they have many different functions that they do. And one of the important functions is they make sure that the neurons don't get too overly excited. So they kind of monitor the space around the neurons. If a neuron gets excited too much, it's activated too much, it can die in a process called excitotoxicity. So the astrocytes, one of their jobs is to take up extra neurotransmitters. So that's neurotransmitters, what's activating your neuron. So it's taking up this stuff out of the extracellular space, out of the fluid surrounding all of the cells and neurons. It's taking up that neurotransmitter that can cause too much excitability in the neuron. It can also help to synchronize neural activity. So if you look at neuron over here and this neuron, the astrocyte can help to make both of the neurons be spiking and active kind of at the same time, instead of being desynchronized. Astrocytes also have a role in establishing the blood-brain barrier, which we'll learn a little bit more about next. Okay, then microglia are the immune system of the brain. They're very interesting cells, and we still have a lot to learn about them because they're difficult to study because they're small and their shapes can change. So they're a little bit fickle cells to be working with. But basically what happens is that anytime you have injury to the brain or inflammation in the brain, the microglia are activated and they will change their shape and their structure, and they can actually migrate through the space between the cells and kind of migrate around the brain, and they can lengthen their processes and spread out so that their synapses can take up things that are potentially harmful in the brain, and then they can also release different types of chemicals to help in the immune system functions inside the brain. And then the last type of glia that we're going to talk about is a Schwann cell, and it's not pictured in this particular image, but Schwann cells have the same job as oligodendrocytes. They also create myelin sheaths. They look a little different from oligodendrocytes, but the big difference is that the oligodendrocytes make myelin sheath in the central nervous system, which is in the brain and inside of your spinal cord. The Schwann cells are making myelin sheath in your periphery. So it's all the nerves that run throughout all your body. Anything outside of your spinal cord is your peripheral nervous system. So the Schwann cells are making myelin out here, and the oligodendrocytes are making myelin in your brain. All right, and then the blood-brain barrier. As you said, the astrocytes do have a role in maintaining the blood-brain barrier. Basically what it is that anything you eat in your body, the chemicals can be digested and they can be passed into your blood through the digestive system. The brain needs a constant influx and flow of blood because it needs oxygen and it needs glucose continuously so that it has the energy to keep firing and keep being active, basically. So the brain, it's very important that it gets a continual inflow of blood, But it's also very important that all the chemicals we take into our body can't all go up into our brain, because as we'll learn, it's kind of it has to have a very particular environment for the cells to function correctly. So it can't just let anything we take into our bodies and all the chemicals we use in our environment and come across in our food. We don't want all those to get up into our brain. So basically, your blood vessels inside of the brain have a specialized type of endothelial cell, and those are skin cells or endothelial cells. And these specialized type of endothelial cells form a very thick wall around the capillaries or the veins that go into the brain. And they're tightly joined to make sure that things can't just pass from the blood out of the membrane around the blood vessels into the brain. So basically we see here, if this is the capillary on the inside, we have the endothelial cell, which tightly wraps around that blood vessel. There are only at small junctions where chemicals and ions and molecules can pass out of the blood system. But then we also have a parasite that's wrapping around the endothelial cell, and then we have these astrocytic end feet. So if you can imagine, there's astrocyte up here, his cell body, and then he's extending his processes with the feet at the end of the process. So the astrocytes are basically hanging out right here. Oops. And so anything that can leak out of this junction between the skin cell can be absorbed into the astrocyte to make sure it doesn't get into the brain. So the good thing is, like we said, the blood-brain barrier protects potentially harmful things that from passing through the blood into our brain. But some of the downfall or the side effects is that it also makes it difficult for drugs that we're trying to use to treat like cancers or tumors or Alzheimer's. It makes it difficult for those drugs to be able to pass from the body into the brain. Because if you think about it, most of the drugs we take are either ingested, or maybe they're injected or put directly into our bloodstream. So the blood is how those chemicals, the drug is getting passed through our body, but the blood-brain barrier keeps those things from getting into the brain. So there are some ways that molecules can get from the blood into the brain. So there are small uncharged molecules like oxygen and CO2 that are small enough to be able to just pass through the endothelial cell into the brain. There are also fat dissolvable molecules that are able to basically, so these are lipid soluble agents that are able to dissolve into the endothelium and come out the other end into the brain. So those are two forms of passive transport. And then we also have active transport. So this can be through transport proteins. So these are special proteins that act like a pore or a channel that allow some molecules to come through. So these include glucose, amino acids, and nucleosides. So glucose, we already talked about, is basically how the brain gets its energy. Glucose is very important, and the brain has to have a constant influx of glucose to maintain brain activity. And then amino acids are some of the building blocks of proteins, and for brain plasticity, for learning and any change or growth in the brain, you do have to have a lot of new protein synthesis in your cells. So the amino acids are very important for proper brain function as well. So there are proteins that allow these molecules to pass into the brain. And then there are also receptor-mediated endocytosis. So there are certain receptors that are going to respond to certain chemicals or molecules. and it will allow those things to pass through the endothelium. So for instance, insulin and transferrin are able to signal through our body and then pass through the blood-brain barrier into our brain. Okay, so 3 different ways, or kind of 4 different ways that molecules can pass from our blood into the brain. All right, so now I would like you to take a minute, pause the video, and you are going to, let me escape here. You're going to open up the slides. And when you open up the slides, you should be able to just directly click and drag on these 4 images. So you're going to match up the correct type of glial cell to its function over here. So you can just click and drag these and leave them. All right, and then when you're done with that, you're going to click over in this box over here. And there's a text box over here, and you should be able to just type your answer to the question. So you'll type it directly into the box. So pause here, and then when you're finished, come back and we'll continue. All right, so we're gonna now focus just on neurons. We talked about the different types of cells in the brain, neurons and the glia, different types of glia, and then the blood-brain barrier. So now we're gonna focus more just on the structure of neurons and how information travels within a neuron. So first thing to note is that neurons are move this around here. Neurons are a specialist type of eukaryotic cell. So it's going to have the same components within the cell body of the neuron as any other type of eukaryotic cell, sorry. So within any neuron, you're going to find a nucleus. Sorry, inside the nucleus, you're going to have chromosomes, and the chromosomes are going to contain the DNA. On the DNA strands are the genes. We'll talk about this probably more in the learning and memory chapter, but to learn anything, you actually have to have new protein synthesis. So there's going to be a part of the genes that will be replicated in the nucleus, and then it will send out mRNA outside of the nucleus, and then it will, inside of the ribosomes, you'll have the formation of new protein synthesis. So you're going to have a nucleus, you're going to have the nuclear membrane and DNA, you're going to have the ribosomes for new protein synthesis. You're also going to have the endoplasmic reticulum. There's smooth endoplasmic reticulum and rough endoplasmic reticulum. And then you have the Golgi apparatus here as well, which will package some of the different kinds of proteins and send them to their destination, other parts of the cell. So same basic parts of a eukaryotic cell are going to be found inside of a neuron. Sorry, one other thing to mention is the mitochondrion. So the mitochondrion are the organelle that creates energy. So it creates ATP for the cell. And energy within a cell is important for many different processes, and we'll find later it's important for generation of an action potential. So just to kind of recap those 3 things, the main things I just want you to remember is that within any neural cell in the cell body, you're going to have mitochondrion producing energy. You're going to have a nucleus which contains the chromosomes, DNA, and then on the DNA are the genes. The genes are going to be replicated or translated within the nucleus when you have any learning event. And then in the ribosomes and the endoplasmic reticulum, you're going to have RNA transcription and creation of new proteins or new protein synthesis. And then another thing to keep in mind, similar to every other type of, or most other types of eukaryotic cells, is that neurons do have a phospholipid bilayer, which creates the cell membrane. So the outer layer surrounding the cell and the dendrites, any part of the cell, it's surrounded by a cell membrane. It's kind of like our skin that's all over us. And our skin keeps certain things from getting into our body, right? So it forms a barrier between the outside world and the inside of our bodies. So in the same way, the cell membrane creates a barrier between the outside of a cell and the inside. And it does this basically to protect the inside of a cell and make sure it can function properly, and to make sure only things that are supposed to be coming in the cell are getting inside of it. And so basically it works because of the phospholipid bilayer. Sorry about that. So the phospholipid bilayer has a hydrophilic head, which means the head is attracted to water. And it has a hydrophobic tail, which means that part of it is averse or repelled by water. And so it makes it very difficult for anything to pass through the membrane, because on one side, that molecule would have to be attracted to water, and then to get through the inside, it would have to be repelled by water. So basically, if a protein could get past this layer, it might get stuck here. So it's a semi-permeable membrane, meaning that it'll only let certain types of molecules pass through. And typically, you need a channel, basically like a pore in the membrane, to allow things to get in and out of the cell. You can have pores or channels that remain open all the time, and these are called leak channels. And things can just come in and out of the cell at will. But most channels are closed, and you have to activate them with something in order for them to open up and let things to cross through the membrane. So the same thing is true of our neurons. At rest, the membranes are generally closed for most types of ions and other types of molecules, and only certain things can pass from the outside to the inside and vice versa. Okay, so then we're going to talk about general flow of information in a neuron. So we know that not everything can just pass through a neuron, but when a neuron is activated, the activation of a cell is going to begin at the dendrites, where these small little branches near the cell body. So you can kind of imagine them like little fingers and hands surrounding the cell body. So information flow is going to begin at the dendrites. Another word for dendrite is tree, so you can think of it like the branching of a tree. Ions that come into the dendrites are going to flow to the cell body, which is the little circular part of the cell, which I showed you in pictures before. So another word for cell body is soma, and soma just means body. And then basically for a cell to become active and pass information to the next cell, it has to generate an action potential. So the action potential is an exchange of electrically charged ions that are like a current of electricity flowing down the neuron. Action potentials are how neurons communicate with other neurons. So if this cell wants to communicate to another cell down here, it has to generate an action potential. The action potential is going to be generated at the axon hillock, which is located along the cell body close to the initial segment of the axon. So it would be somewhere over here. Okay. Once the action potential is activated, it's going to travel down the length of the axon until it gets to the presynaptic terminals. The other name for axon is axis. The myelin sheath that surrounds the axons helps the action potential to travel faster. There are cells that do not have myelin sheath, and they still can transfer information, but the action potential will travel a little bit slower. The nodes of Ranvier are little gaps in between the myelin sheath, and basically those help to boost the action potential and make it kind of jumps it from one spot to the next. So it'll jump the action potential from one node of Ranvier to the next, and we'll talk about that more in detail later in the next lecture. And then so once the action potential travels all the way down the axon, it will reach the presynaptic terminals, and it should cause release of neurotransmitter from the terminals. When neurotransmitter is released from here, if there's another cell here with its dendrites over here, the neurotransmitter could activate this cell and cause another action potential. It's not a great cell, but that's the idea. So that's the basic flow of information in a neuron and the basic parts of a neuron, typical neuron. Okay, so again, I want you to pause the video here and open up the slides. And you're simply just going to click on these boxes here and drag the box to the appropriate labeling area. So pause the video and finish that part. All right, so now we're going to go back and do a little bit more in-depth information about each one of these steps. So the first step we're going to go over again is information flow starts at the dendrites. Oh. All right, the dendrites out here. And so these two images are, this is an actual image, a very close-up image of two synapses. So this will be a presynaptic synapse, or pre-synapse, and a post-synapse. This is the axon, and this is the dendrite here. So imagine there's another cell with its axons contacting the dendrites of the cell. Okay, so axon and the dendrite, and the way you know this is an axon is because it has these small circular structures inside of it. These are the vesicles, and the vesicles contain neurotransmitter, and when the cell is activated, it's going to release the neurotransmitter into this gap between the two cells. You can kind of see the gap here. And then the neurotransmitter will bind to the postsynapse on the dendrite and activate this cell. So one thing to keep in mind is we didn't used to have the technology we do now to be able to see at this type of a scale in this picture here. Early on, there was debate between researchers whether or not cells communicated through chemicals, which would have this gap between cells and release the chemicals between the gap, or whether or not cells were talking to each other through electrical activity, in which case they would be directly connected to each other and just passing that action potential directly from one cell to the other because they would be directly connected. So with time, researchers were able to demonstrate that, in fact, cells are communicating through chemicals, which we now call neurotransmitters. There are other types of chemicals as well, but we'll just focus on neurotransmitters for now. Over here, we see another way of looking at this. So obviously, this is a cartoon. So we see these vesicles inside of the presynapse, which is the axon terminal bud. If this cell were activated, it would cause the vesicles to bind with the membrane on the synapse, and it would spill out its contents into the synaptic cleft, which would be neurotransmitter. So neurotransmitter would flow into the synaptic cleft or gap, and then the neurotransmitter would bind to receptors on the postsynaptic side. So again, receptors are kind of like the pores in the membrane, and things can't pass into the cell unless the receptors open up. At rest, they're going to be closed, and when the neurotransmitter binds, they'll change their shape. I can't really do it with my hands, and they'll open up to allow things to pass inside. So a neurotransmitter will bind to the postsynapse, and this allows ions to enter into the cell. So just remember that the neurotransmitter that I put in black is opening the cell, it's binding to the receptor and opening it up, but what's actually passing into the cell are ions. And we'll talk about ions a little bit later in the lecture. Okay, so this video kind of demonstrates for you what it might look like when a receptor opens. So I'll explain the outside of the shape here is this whole portion is the receptor. This white portion on top, the green portion, and these long little things. And so you can see at rest, this is the membrane on the bottom here. So at rest, the membrane is totally closed. And then there's these sort of slots in the side of the receptor. And these are binding sites for the neurotransmitter. So each receptor is going to have very specific binding sites that will only allow certain neurotransmitters to bind. So let's just look and see what it looks like when the neurotransmitter binds. Okay, so basically saw when the glutamate, the neurotransmitter, came into the little slot and bound to the receptor, it caused this bottom portion of the receptor to open up a hole in the membrane, and allowed the ion sodium to pass into the cell. So that's just to give you a little visualization of what an action or what a receptor opening might look like. So go ahead and, if you're feeling a little overwhelmed, there's a lot of new information, take a quick 5 or 10 minute break here, pause the video, and come back. Okay, so we talked about receptors opening. I'll just draw a little receptor here. Okay. On the dendrite, on the postsynaptic side of a cell, and when it opens, it allows ions to pass through. So now we're gonna segue and we're gonna talk about ions a little bit, and then talk about how they set up a neuron to be able to generate the action potential. Okay, so ions are molecules that have either a positive or a negative charge. They can be anions, which are negatively charged, or cations, which are positively charged. And in the brain, the major ions that we're going to be talking about are sodium, potassium, chloride, and calcium. And most of the time I'm going to be using the short form of the ions. Okay, and so in order to have an action potential, there are two properties that act on the ions to create the necessary environment for an action potential to take place, basically. Okay, so we have what's called electrostatic pressure and diffusion are the two properties that are setting up a neuron to be able to have an action potential. Okay, electrostatic pressure is just saying that two ions that have the same charge, two cations or two anions, are going to be repelled by each other, but two ions that have the opposite charge are going to be attracted to each other. So chloride and calcium might be attracted to each other. Sodium and potassium, or sodium and calcium, would be repelled by each other. And then diffusion is a natural tendency for ions to want to spread out or disperse over a space. So if you have, we'll see a demonstration of this, but if you have a cup with some water and you place some dye in the water, it's going to be highly concentrated in one area at first, but then over time, it's going to evenly distribute itself throughout the entire mixture. And you can see that by just putting dye in some water. So we'll look at a video that explains electrostatic pressure a little bit using air bubbles and a charged water bottle.
Hi folks, here's a quick fun science experiment that demonstrates a bunch of electrostatic stuff with soap bubbles. Start by finding a very smooth surface. The smoother the better. A smoother surface helps the bubble keep its shape. I'm using this laminated tabletop. Next, mix up some dishwashing soap and water. Lastly, get a drinking straw. To blow a large bubble, suck some soap into the straw, but not into your mouth, and then blow it gently onto the smooth surface. The surface needs to be coated in soapy water to form the bottom of the bubble. Now for the real fun. We need an electrically charged object. We can use the triboelectric effect to charge a variety of different things. Try rubbing a balloon or a plastic Coke bottle against your hair to charge the balloon or Coke bottle. A lot of different things will work. See my video about the triboelectric effect for tips on this. Bring the side of the object that you charge near the soap bubble. The soap bubble is attracted to the charged object. If you come too close, it'll burst. Have fun pulling the soap bubble around or making it change its shape. Next, blow a smaller bubble in the center of the bigger one. Notice that when you attract the bigger one around, the smaller one doesn't move. Why not? Here's an explanation for all this. When you charge up an object, depending on the material you used, the area you charged either has more positive protons than negative electrons, like this glass, or more negative electrons than positive protons, like our plastic bottle. For our bottle, we say it's negatively charged. The soap bubble overall is electrically neutral. It has an equal number of positive protons and negative electrons. But it does contain impurities such as metals that are ions, i.e. they're charged either negatively or positively, and the skin of the bubble itself is charged. So while overall the bubble is neutral, the charge within it can move around, like with a metal object. With the soap bubble shown here, when you bring a negatively charged object close to the bubble, the negative electrons on the object attract the positively charged ions within the bubble, even some from the other side. That's because opposite charges attract each other. Now you have one side of the bubble with more negative charges than positive. We say it's negatively charged. And the other side has more positive charges than negative. We say it's positively charged. And just as like charges repel each other, opposite charges attract each other. So the negatively charged bottle attracts a now positively charged side of the bubble. It also still repels a negatively charged side of the bubble, but that's further away, so that repulsion is weaker. The attraction between the negatively charged bottle and the positively charged side of the bubble is stronger and wins. The bubble moves towards the bottle. And what about our smaller bubble that didn't move? It wasn't attracted.
All right, if you want to finish the video, you're welcome to pull up the slides and keep watching it. Okay. All right, so then when we're talking about electrostatic pressure with a neuron, what's happening is that the ions are not equally distributed on both sides of the membrane. And so we actually have the inside of a neuron is actually going to be more negative compared to the outside. So if the inside's negatively charged and the outside's positively charged, it might look something like this, where negatively charged ions are going to line along the inside of the membrane, and the positively charged ions are going to line around the outside of the membrane. And that way they're kind of ready. If the membrane were to open, have a hole in it, then the ions could kind of push through the membrane towards the negative or positive charge. And we would say that the inside of the cell is negatively charged compared to the outside of the cell. Oh, and the charge that we're talking about, I was trying to understand, what did this mean? The charge, the thing that creates the positive and negative charge in our cells is the distribution of ions, because they have those positive and negative charges to them. So they are creating the electrostatic pressure. Here's a video about diffusion.
On cells and on cell biology. So to get started, we're going to talk a little bit about diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So to demonstrate how that works, we're going to do an experiment in which I'm going to drop a drop of food coloring into a beaker of hot water and another drop of food coloring into a beaker of cold water at the same time. So I'm using red food coloring to demonstrate the hot beaker and blue food coloring to demonstrate the cold beaker. So we're going to be looking to see how quickly the food coloring spreads out in the two different beakers. So here we go. Now, as you can see, the red food coloring is moving through the beaker of hot water much more rapidly than the blue food coloring is moving through the beaker of cold water. And that's because diffusion is based on molecular movement. Molecules will tend to speed up at a higher temperature, which means that diffusion will happen more rapidly. Now, how is this important in the context of the cell? Now, in a cell, diffusion can happen across the cell membrane. The cell membrane is what we call a semi-permeable membrane. And what that means is that some molecules can flow freely across it or diffuse freely across it, while others will be blocked or stopped from crossing. One of the most important criteria that we use to determine whether a molecule will be able to cross is size. So small molecules can cross freely across the cell membrane, whereas large molecules may be blocked from diffusing across the cell membrane. So we're going to do an experiment that's going to demonstrate.
All right. So you can, don't worry too much about what she was saying about temperature. Temperature it is important for our brain function, but just pay attention to this idea of diffusion. So if you have, if you put any type of a molecule or ion or protein into a solution, it's going to want to diffuse or spread out evenly within that solution. So diffusion acts on ions. Let me put it back in presentation mode. Diffusion acts on ions when there's a difference in the concentration of ions across the membrane, and then we call this concentration gradient. Okay, so for instance, when a cell is at rest, meaning when the neuron is not firing and being active, it's just hanging out, there's more sodium outside of the cell than inside. This means the concentration gradient is pushing sodium into the cell because it wants to be evenly distributed on both sides of the cell. For potassium, there's more potassium inside the cell than out. So the concentration gradient pushes potassium outside of the cell to evenly distribute. And then for chloride, there's more chloride outside the cell than inside. So the concentration gradient pushes chloride to be in the cell. Calcium is the same as sodium and chloride. There's more of it outside the cell than inside, pushing calcium into the cell. Okay, so we've talked about how the concentration gradient is working to push the ions into and outside of the cell. So what about electrostatic pressure? And first we have to understand, we already talked about it a little bit, but the inside of the cell is negatively charged compared to the outside of the cell. It is negative 70 millivolts. And this is basically if you were to measure the voltage outside of the cell and then measure the voltage inside of the cell, and you compare relative to each other, the inside of the cell is negative 70 millivolts. This is because there are large organic anions. They're too large to pass through the cell membrane. So kind of like the video was saying, not all types of molecules can pass through the membrane. If they're too big, they can't pass. So we have a higher concentration of we'll put that back. Okay, we have a higher concentration of these organic anions inside of the cell than outside, which is making inside negative. So when the cell is at rest, the resting membrane potential is negative 70 millivolts. That's what we call it, the resting membrane potential, or it's basically the electric potential that exists between the in and outside of the cell. So it's called the resting membrane potential. So this means that any of the positively charged ions are going to be attracted to the inside of the cell based on electrostatic pressure. So having the cell be negative 70 on the inside sets up electrical gradient. So the positively charged ions outside of the cell are lining up along the membrane outside, and the negatively charged ions inside of the cell are lining up along the membrane on the inside, setting up an electrical gradient between the in and the outside of the cell. As I already said, because the inside is negative, that means that positively charged cations are going to want to move into the cell. So then to put this all together, we have sodium, potassium chloride, calcium, the concentration gradient, so how many ions are outside versus inside. Concentration gradient pushes sodium inside because there's more sodium out than in. There's also more chloride outside than in, so it wants to go in, and there's more calcium outside of the cell than inside, so it wants to go in. But there's actually more potassium inside of the cell than outside, so it wants to go out. Okay, so that's the concentration gradient pulling on the ions. The electrical gradient is basically I'll go back to over here. Any of the positively charged ions are going to want to move into the cell. So sodium, calcium, potassium want to move in the cell, and the negatively charged ions are going to want to move out of the cell. So chloride wants to move out. So what you end up with is chloride and potassium have the two different properties are working against the ions in opposite directions. So they're being pushed and pulled in opposite direction based on the two properties. However, sodium and calcium are both being pushed into the cell based on the two properties. So both properties are working in the same direction. So sodium and calcium are both being pulled into the cell. But essentially, calcium, it is a very important ion, and we'll talk about it later on in the class. It has some very important functions in the cell, but it doesn't have a role in generating an action potential because there's so little of it. So if you compare like how much potassium there is, 135, how much sodium there is, would be actually 140 and 168 versus 1.2 for calcium. So it's just very, very small numbers, small amounts of calcium, so it's not quite enough calcium to change the voltage of the cell very much as calcium goes in and out of the cell. So basically, the main type of ion that's going to be starting or triggering an action potential is sodium, because it has both of those properties pulling sodium in the same direction. So potassium gets kind of canceled out, chloride gets canceled out, and calcium is like there but kind of canceled out. Okay, so we're gonna go back when we start the next set of lecture series. We're gonna talk more in detail and put this together about generating an action potential. For right now, we're just going to leave it there, so we know that these ions are forming the basis of an action potential, and that when we look at all the property of electrostatic pressure and the property of diffusion, sodium is being driven into the cell. The other cells are kind of maintained at a kind of homeostatic or even level because of the two properties. So we're going to kind of think about and concentrate on the role of sodium. And then the next question could be, so why don't the ions just rush in and out according to these forces? Why don't they just even themselves out? And we're going to go back to this idea of the semi-permeable membrane, or selective permeability. So the cellular membrane has selective permeability, meaning some chemicals can pass more easily than others, which we've already covered a little bit. We're going to talk about specific types of receptors that can let the sodium ions in. Again, we need to know this so we understand what's happening during the action potential. We already talked about, you can kind of see here, so there are some types of channels which are leak channels that are just open all the time and can let ions pass in and out of the cell anytime at will. But for the most part, most channels are closed when the cell is at rest, and they need to be opened by some type of a force. So what I would like you to do now is just to kind of get more of a visual, you will go to this, you'll hit Control-click and follow this link, and it'll take you to a simulator. I'll go over the simulator at the end of the lecture just so it doesn't get messed up. So you'll go to the simulator, and I want you to play around a little bit with leak channels and then channels being activated. But for right now, we'll go back. So we talked about most of the time, channels have to be acted on for them to open up and let things to pass through the cell. The ions have to pass through channels or gates to get into this cell. These are closed when the cell is at rest, and there are different ways that the channels can be opened. So far we've talked about neurotransmitter being released from the presynaptic synapse and binding to the postsynaptic synapse. And when it binds, it's actually going to change the shape of the synapse because proteins and molecules are kind of sticky and they attract and detract. So when you add like a new chemical or a new molecule, it'll change the shape of a protein because things kind of stick and move around in relation to each other. So you add this chemical, the neurotransmitter, it's going to change the shape of the receptor and allow the ions to pass through. This is what we covered already. This is called a ligand-gated receptor. And this is you can think of it as the lock and key system, where every kind of neurotransmitter can't open every kind of receptor. Receptors have specific neurotransmitters that will open them, and the area where the neurotransmitter binds is only going to, it's kind of like like a lock and key, where the shape of the receptor site has to match the shape of the ligand. So that's why it's called a lock and key system. Okay, but besides just the ligand gated, channels can also be voltage gated, which means that when the voltage of the cell on the inside or the outside changes, the receptor will open up. This is what's going to happen to generate our action potential. And it's going to be a voltage-gated sodium channel. So when the voltage of the cell changes, the channels open up and they let even more sodium come in, which we'll learn about in the next video. And then we also have tension-gated receptors. So these are more often found maybe in the skin and the periphery. But basically tension or pressure can cause like a bending in the membrane, and it can physically cause the receptor to open up. So maybe if you touch this receptor, it bends the membrane and pulls these two sides of the receptor out. And then it will open up the receptor and allow ions to pass through. So different types of receptors, the voltage gated, the ligand gated using neurotransmitter, or the tension gated. Okay, and then we talked about receptors. We talked about leak channels. There are also pumps. So the pumps are going to have to use energy from ATP, which comes from our little mitochondrion in our cells. The energy from ATP is used to run a pump. Okay, and so one of the very important pumps inside of a neuron is called the sodium-potassium pump. And basically, we talked about how there's more sodium outside, O-U-T, and more potassium inside the cell. And these cells, the ions are kind of being pushed against their concentration gradient. And in the case of potassium, they're being pushed against the electrochemical pressure gradient. So basically, this pump is going to take sodium from inside the cell, and then it's going to use energy, which is depicted as the little red thing. It's going to use ATP to pump the sodium outside of the cell. And then when it opens up to the outside, now the potassium can come in and bind to it. And then it opens back up to the inside and dumps the potassium out on the inside of the cell. And so basically it pumps 3 sodium ions out. I'll just put this for out. And 2 potassium ions in every time it's activated. Using energy every time it does that to set up that concentration gradient that we talked about, where there's more sodium, 3 out, and less calcium, more sodium outside the cell. So it's setting up that concentration gradient that we need. All right, so that's the sodium-potassium pump. And that essentially concludes the information we're going to cover for today. We've covered some of the basic types of cells, the basic anatomy of a neuron, basically how information flows in a neuron. And then we've gone in a little bit of detail about what sets up a neuron to be able to fire an action potential, which is having that semi-permeable membrane, having electrostatic pressure and diffusion, working on ions, and the sodium-potassium pump, which is setting up the concentration gradient of sodium and potassium. So all those things are happening just for a cell to be able to have an action potential. So go ahead and, again, stop here, stop the video, pull up your slides, and please fill out the slides. So for each ion, you're going to type in here whether or not the concentration gradient is pulling it in or out of the cell, and then whether or not the electrical gradient is pulling it in or out of the cell. You're going to click on the arrow and drag it up and down to line it up with whichever ion has both properties pulling it in the same direction. And then here you're going to do free response, click in the box, and you're just going to type in your response to the question here. All right, so that is it for today. For the quiz, I have not fully decided if I'm going to do a quiz here. So for general, for being able to study for the class, there are different things that you can do. If you are, I'm the kind of person, I like to hear the lecture and I also like to read the material. So I do highly suggest actually reading your book. It has a lot of very good information in it, and I can't possibly cover everything in the book in the lectures as well, because it would be really, really, really long. So please read your book. It's really well written, and then it has a lot of what it calls stop and check as you're reading. There are little sections called stop and check with questions. So I suggest that you do those. And at the end of each section, there is a multiple choice section. And there are what they call thought questions. So I would highly suggest doing that little multiple choice quiz. Some of the questions I will use in maybe tests or quizzes, and it will also help to cue you in on what the important information is. So if the book finds that information important, you can pretty much guess I'm also going to find that information important, and it's probably going to show up on a quiz or a test somewhere. So using those little quiz sections at the end is a good way to study. You can also go to YouTube. On YouTube, there is Khan Academy, which is really great. It's more slow-paced videos, so it's great for learning new information. And I've done several courses on Khan Academy. I think it's great. There's also Crash Course. Crash Course gives you more cartoons and visuals, so if you're a highly visual person, I would suggest Crash Course. Okay, my finger sticks and then I can't write, but okay, Crash Course is going to be more fast-paced though, so if you're learning something for the first time, it might be hard to follow these videos. So there's cons and pros to both, and then there's also Quizlet, which will have Quizlet is information that other people have created. Sometimes teachers create it, sometimes students create it. So you might have flashcards on there, you might have diagrams, little quizzes and things like that. For any of these, you would just search for maybe action potential or brain cells. No, I'd probably just stick with action potential and then look for Khan Academy, Crash Course, or Quizlet, and it should give you some extra resources to help you in your studying if you need something beyond the book in the lectures. Okay, so that's everything for today. Thank you very much. Hopefully I have not overwhelmed you too much, and have a great one. I'm gonna hit stop share and stop recording.